Welcome. In this session, firstly, I'm going to discuss about biochemical characterization of transforming principle. Then later, we will see how scientists unequivocally prove that DNA is the genetic material. And then later, I'll also start discussing about DNA versus RNA. So let's begin this session with biochemical characterization of transforming principle. In the last session, we understood Griffith proved the transforming principle, and when he took S train and heat killed and mixed with the live R strain, he saw that the R strain transformed into S strain. Okay, the R strain transformed into S strain, and he concluded that something is taken up by this R strain from the S strain and it got converted to S strain. But what is that? We know here the organisms are mostly composed of molecules like proteins, carbohydrates, lipids, nucleic acids. Now, out of these major molecules that we find in living organisms, which one passed from S strain to R strain and converted this R strain to S strain? So that was a question. Now, to solve this, three scientists, Oswald T. Avery, right, and another person, Colin MacLeod, and another person, Macklin McCarthy, are three scientists, okay, who did experiment to find out of these proteins, carbohydrates, lipids, and nucleic acids, which one passed from the S strain to R strain and converted the R strain into S strain. Okay, so these three, of course, together we call it an Avery experiment. It's called Avery experiment. Okay, or OT, Avery experiment also you can call it. And let's see this experiment. I'll simply mention it OT Avery experiment. Now what they have done is to see which of these molecules passed from, okay, the S strain, that is heat kill, to R strain, right? So they have taken again the S strain bacteria, so that is heat killed and homogenized. Okay, so which means they ground it and then they mixed this one with the enzyme called carbohydrases. So right, the enzyme, so these carbohydrases is a collective word that consists of different types of enzymes which will act on carbohydrates and digest them. So when they added carbohydrates to this S strain, that is homogenized, right, in this case it is going to remove the carbohydrates from the S strain. So it results in removing carbohydrates from S strain. it results in removing carbohydrates from S train. So now, right, this one, after okay treating with the carbohydrates, they added this okay S strain. So S strain that is treated with carbohydrates, which means that this S strain is not having carbohydrates. Now they mixed this with the live R strain, and okay, what they observed is that, so after doing this, they saw that, right, R strain was converted into S strain. So conversion took place. So actually, if carbohydrates are the transforming agents, as I have removed here, this conversion, okay, the R strain to S strain should not take place. Okay, but here we see it has taken place, and this makes us to understand that carbohydrates are not the transforming agents. So with this, it's confirmed that carbohydrates are not transforming agents, is not transforming agent, right? So once again, so S strain that is treated with carbohydrates is, which means this S strain is not having carbohydrates, and they mixed with live R strain, and what they observed is that the R strain converted into S strain, right? If the carbohydrates are the transforming agents, this conversion should not take place because the carbohydrates are not present in the S strain, right? And but here it has taken place, and this makes us to come to a conclusion that carbohydrates are not transforming agent, right? So R strain to S strain conversion occurred, so in this case, right? So with this we eliminate the carbohydrates. So carbohydrates are not the transforming agents. Now let's look at with the others. Okay, so again I'm going to take in this case same S strain, that is heat killed and homogenized, but now I'm going to add proteases. So proteases are enzymes that digest proteins, so that digest proteins. Okay, so with this what we see here is S strain doesn't have proteins, so all those proteins are removed. Okay, now I'm going to mix this S strain where okay proteins are removed, and then mixed with the live R strain, and when the results okay came up, what we have seen is that R strain again converted to S strain, so that is conversion occurred, right? Okay, but if the proteins are the transforming agents, this conversion should not take place, but it has taken place, and this makes us to understand that proteins are also not transforming agents. So they are also not transforming agents, so we are going to okay remove these proteins okay from the list. Now we will see again this experiment with the lipases. So I'm going to add lipases. So in this case, the lipases will digest lipids, and that's what we get. The S train is without lipids. Now I'm going to take that S train, so that doesn't have lipids, so without lipids, is mixed with the live R strain, and then what we see here again, the conversion of R strain into S strain occurred. Okay, so in this case also conversion occurred, right? And with this we come to a conclusion that lipids are also not transforming agents. So they are also not transforming agents, right? So if the lipids are transforming agents, then this conversion should not take place because the lipids are absent in S strain. Okay, and with this we eliminate lipids also from the list. And then coming to the nucleic acids, we know there are two types of nucleic acids, that is RNA and DNA. Now I'm going to take enzyme called RNases. So this enzyme RNases, okay, so these are enzyme, so this is an enzyme that digests RNA. It means it will remove RNA. So what we get here is a strain that doesn't have RNA, so that is without RNA, so it is not having RNA. So now I'm going to again mix this S strain, which is without RNA, to a live R strain, and what I observed, okay, still the R strain converted to S strain. So again conversion occurred, and with this we conclude that RNA is not the transforming agent, right, or the genetic material. So we know that genetic material will pass, okay, here from the S strain to R strain and is going to convert that R strain into S strain. So here RNA is also not the transforming agent. Now lastly, so with this I'm going to remove RNA also. So RNA is also not the transforming agent. Now lastly, right, so they added DNases. So these are enzymes, okay, enzyme that digest DNA. So now what we obtain is a strain without DNA. Now, so those three persons, they mix this S strain that is without DNA to a live R strain. So in this case, what they observed is that the R strain remained R strain. It did not convert into S strain. Okay, so that's what conversion did not occur. So conversion, okay, so did not occur. And with this, they concluded that DNA is the transforming agent. So DNA is the transforming agent, right? and out of all this they eliminated proteins, carbohydrates, lipids, RNA, and they found that okay, DNA is the transforming agent or the genetic material. But many scientists did not agree because uh, in the living organisms the majority of the component present is proteins. So all the scientists they thought it's the proteins which are the transforming agents, right? And now there are other scientists who did experiment, okay, and they finally proved that DNA is the genetic material. So let's see about the experiment. It's called Hershey and Chase experiment. Called Hershey and Chase experiment. So they unequivocally okay proved that DNA is the genetic material. So this experiment proved that DNA is the genetic material. So I already told you that many scientists have voted proteins because the majority of molecules that exist in living organisms are proteins, and they thought the proteins are the transforming agents or the genetic material, right? And with this experiment, okay, it was finally concluded and there is no doubt whether the protein or the carbohydrate or the DNA or RNA is the genetic material. So once for all we came to conclusion that DNA is the genetic material, okay, with this experiment, that is Hershey and Chase experiment, right? So two scientists, one is Alfred Hershey, right? So this person is an American bacteriologist and genetist, along with another person named Martha Chase, right? So both of them they confirmed the genetic material is DNA in 1952. In 1952. So whereas uh, the OT Avery, so they have done it from 1933. So here OT Avery's experiment. So that was from 1933 to 1944. So it was in 1933 to 1944. After them, okay, Hershey and Chase, they performed this experiment, okay, and that is in 1952, and proved that DNA is the genetic material, right? Now the scientific world have a question, is that whether it is protein or DNA genetic material. So which one of these two is the genetic material? So whether it's the protein, because some are saying that protein is the genetic material, whereas OT Avery and his colleagues, they proved that DNA is the genetic material. So the question is to find out of these two, which one is the genetic material. Okay, now to find out this, Hershey and Chase, what they have done is that they have grown some viruses on a medium containing radioactive sulfur. Okay, so we'll see that one. The proteins, what these are made up of. The proteins consist of carbohydrates, hydrogen, oxygen, nitrogen, and sulfur is present in proteins. Whereas DNA does not have protein. Instead, it will be having all the same components. It will also have carbon, hydrogen, oxygen, nitrogen, but instead of sulfur, it will have phosphorus. So S here is sulfur and P is phosphorus, right? There is no protein that contains phosphorus and no DNA will have sulfur. Okay, and now what they have did Hershey and Chase, they have taken bacteriophages, right? So they have taken bacteriophages. So bacteriophages that infect E. coli bacterium. So that infect E. coli bacterium. We know that bacteriophages are viruses. So they are viruses, right? So the bacteriophages that infect E. coli, so they have taken, and now they have taken two mediums, right? So this is a medium containing radioactive, okay, sulfur, and this is medium containing radioactive phosphorus. Okay, so they have taken radioactive sulfur and radioactive, right, sulfur. So here the radioactive phosphorus, what they have taken is P32. The radioactive phosphorus they have taken is P32, and the radioactive sulfur that they have taken is S35, right? Right. So here is the medium containing radioactive phosphorus, that is P32, and the radioactive sulfur, that is right, S35. Now what they have done is they have grown bacteriophages in this medium. So grown bacteriophages for several generations. Okay, and here also they have grown some bacteriophages. Right, some bacteriophages they have grown in medium containing radioactive phosphorus, and some other bacteriophages they have grown in medium containing radioactive sulfur. Now what happens to those bacteriophages which are growing on this radioactive phosphorus? So let's see it. So we know the bacteriophage, okay, structure. So I'm going to take, right, like this. So this is a bacteriophage growing on medium containing radioactive phosphorus, that is P32. So this is grown on medium containing P32, right? So in this case, we know that, as I told you already, I will just write again here. So protein that consists of carbon, hydrogen, oxygen, nitrogen and sulfur, and in case of DNA it has same components except phosphorus, right? Now the bacteriophages that are grown in the radioactive medium containing radioactive phosphorus, okay, what they have seen after several generations, its DNA became, right, radioactive. So in this case, this is DNA which is radioactive, so it is having radioactive phosphorus. Okay, now when we see the bacteria that is grown on radioactive sulfur, in this case the DNA is not radioactive, instead the protein coat became radioactive, right? And its DNA is not radioactive. So DNA is not radioactive, but the protein coat is radioactive. Right? I hope you find the difference between. I'll just mention here, grown on S35. So already I told you that this thing you have to keep in mind. The protein contains sulfur, whereas that sulfur is not present in case of DNA, whereas DNA has phosphorus. Okay, now when we have grown the bacteriophages on medium containing phosphorus, it will have DNA that will be radioactive, and the bacteriophages that are grown on medium containing radioactive sulfur will have the protein coat that is radioactive. So in this case, this protein coat is not radioactive. So this protein coat is non-radioactive, and in this case DNA is non-radioactive. It's not radioactive, right? So now what they did, they allowed these bacteriophages to infect E. coli. So I already told you the bacteriophages will be infecting E. coli. So let's see. So these are E. coli bacteria, and they allowed these bacteriophages to infect them. And let's see here. So the bacteriophages are going to attach, okay, to the E. coli, and they are going to inject the nucleic acid into this, whereas the protein coat will never enter into the host cell. So we need to know about the life cycle of a virus. So how does viruses will be infecting the host cells, and what is all its cycle. Actually, when a virus attacks a host cell, its protein coat will not enter the host cell. The protein coat remains outside, and what enters into the host cell is its genetic material. So that is its DNA entered, and in this case, right, we'll see here. So in this case, again the same, the genetic material entered. Okay, so this is DNA which entered into the host cell, but it is non-radioactive, and in this case it is radioactive DNA. So this is radioactive DNA, right? And this step is called infection, right? The next step will be blending. So what they are going to do in blending, the viral coats they are going to remove from the bacteria by agitating them in a blender. So in this step, okay, so they will remove viral coats. So these are the viral coats. They are going to remove these viral coats from bacteria. Viral coats from bacteria by agitating them in a blender. So it's a grinder, Okay, the blender is a grinder, so which will help in separating these viral coats from the bacteria. Okay, so by agitating them in a blender, so that step we call it blending. And lastly, the third step will be centrifugation. The last step will be centrifugation, and this step will help in, so it helps in separation of, so this step is going to help in separation of virus particles from bacteria. So the centrifugation step will help in separation of virus particles from bacteria. And we are going to get, okay, so here there are two test tubes. So you can see two test tubes here. And after centrifugation, what we are going to see in the test tubes, residue and supernatant. So what you see here on the top will be, okay, supernatant, and below that we are going to see the residue. Right, so here, so this is supernatant and this is the residue. Okay, the supernatant will be having the virus particles. The supernatant will have virus particles, and the residue will be having, okay, E. coli cells. So they will be having E. coli cells. Now, okay, the test tube that is a centrifuge with having E. coli, okay, that was infected with the bacteriophage which was grown on radioactive phosphorus P32, and what they have seen, okay, the supernatant was, okay, radioactive. So it means that it is having, okay, so radioactive, okay, sulfur. I'm sorry, I have mentioned this one, but I just mentioned. So this is the test tube, which, okay, so in this case it's having radioactive sulfur in the supernatant. And what we see in the case of residue, in that case, okay, no radioactive detected, okay, in the residue of this test tube, okay, which I have taken from, okay, E. coli that were infected with bacteriophages which are grown on radioactive sulfur, that is S35. Okay, so actually I mentioned earlier this, I'm sorry, so this I need to take, so is the test tube for this one. And for this one, okay, this I'll mention here, okay, here in this test tube. So here the E. coli bacteria that were infected with the radioactive phosphorus, right, so is the test tube here of and when we have seen it, the virus particles, because in this case the virus coat, so this is the virus coat, the virus coat is not radioactive, so it is not radioactive, only its DNA is radioactive, which I have already mentioned. And in this case the virus code, that is protein code, is radioactive. The virus code is radioactive. So when we see this one, right, the residue that has E. coli cells, you can see the E. coli cells are having the radioactive DNA inside the E. coli cells, and in this case the residue is not having of course the radioactive DNA, right? And in this case, so this is, I just mentioned, in this case E. coli cells without radioactive DNA, and in this case E. coli cells with the radioactive DNA. So with P32, so that is with the P32 in this case, and here the supernet will be having S35. So with this experiment we come to a conclusion that it is the DNA that passed from this bacteriophage into the E. coli, right? And with this it's clear that it's the DNA which is a genetic material. So DNA is the genetic material. So with this experiment it was proved that DNA is the genetic material, right? I hope it's clear about this experiment. Okay, and finally we understood that DNA is the genetic material, not the protein, right? So next we'll see about properties of genetic material. So that I'm going to discuss between DNA versus RNA. Already in the beginning I have mentioned, okay, properties of genetic material. To be a genetic material it should have some properties like able to generate its own replica. So it should be able to replicate by itself and should be stable chemically and structurally, and should provide scope for slow changes, right? And should be able to express itself in terms of Mendelian characters. So those are the things which I have mentioned, okay, in the first sessions, okay, of this chapter. Now let's see here uh, the differences between RNA and DNA. So we have to keep in mind, so what is the first genetic material? So the first genetic material. So here what we believe is that the life on this planet Earth evolved itself, okay, somewhere in the waters, okay, and we do not know how the first self-replicating molecules evolved, okay, that is a still question. And okay, long back the first replicating self-replicating molecule that developed was actually RNA. So hence I say the first genetic material is RNA, and in course of time this molecule RNA, which is less stable, okay, undergone a lot of chemical changes and resulted in the formation of much more stabler form, that is DNA. So what I say is that in course of time RNA evolved into DNA. So that is RNA evolved into DNA, okay, and the RNA is less stable and when compared with the DNA. So and so this DNA is much stable when compared to RNA. So how is that RNA is less stable and DNA is more stable? So we will find out in the differences. So let's see those differences, DNA and RNA. right? So let's see, DNA double stranded is usually double stranded, but there are some instances where we can see the DNA as a or in single stranded form. And coming to RNA, this is usually single stranded is usually single stranded, but it can also become, right, double stranded. Then coming to the pentose molecule here, so there, in case of DNA, okay, the second OH group. So I told you while discussing the pentose molecule, so the pentose molecule in case of DNA, it doesn't have the second OH group, that is hydroxyl group, right, is absent. So the second OH group is absent in the pentose sugar molecules, right? And this makes, okay, it to become less reactive. But in case of RNA, so the second OH group, that is hydroxyl group, is present in the pentose sugar. So of course that's why it's called the ribose sugar, and in this case it is called deoxyribose. So absence of this second OH group actually makes, okay, DNA less reactive. Okay, so this helps DNA to become less reactive, while the presence of the second OH group actually helps RNA to become more reactive. So it will be more reactive due to presence of the second OH group, okay, in case of pentose sugars here. Then, right, coming to the DNA, here DNA is chemically less reactive and structurally more stable. So it is chemically less reactive, which I already mentioned, and structurally more stable. So which is opposite in case of RNA. So here RNA is, okay, so it is catalytic. So in some cases, okay, it will act as a, right, catalyst, right, and hence, okay, and hence it is more reactive. Okay, so it will be acting as catalyst. That later I will discuss what are the RNAs that act as catalyst like, okay, and they will be more reactive. Okay, so DNA is less reactive and structurally more stable here. So there is no catalytic function of DNA. So no catalytic function. So DNAs do not act as catalyst, right? Then, so we have here is, okay, presence of thymine instead of uracil. So this also makes the DNA to become, okay, structurally more stable. And in the case, okay, here there is in the RNA there is presence of uracil, okay, and this also makes it more reactive. So the presence of uracil, so we know of course the same uracil, okay, undergone modifications by addition of methyl group at okay, fifth position, and it became thymine. So that's why I mentioned in our earlier sessions thymine as 5-methyl uracil. Next, DNA mutates at slower rate. So it mutates at slower rate, whereas in case of RNA it mutates at faster rate. So the mutations are seen very much frequent in case of RNA, right? Whereas in case of DNA, as it has two strands, the process of mutation will be much slower when compared with the RNA, which has only one strand. Then the DNA will be dependent on RNA for the synthesis of proteins. So DNA depends on RNA for synthesis of protein. So how it is going to depend on RNA for synthesis of proteins, that I will be discussing at the right point where I'll be discussing synthesis of proteins, that is a translation, transcription and translation. So while discussing that, I will be telling you how DNA is going to depend on RNA, how it takes that help. And in case of RNA, so this RNA directly codes for the synthesis. So RNA directly codes for synthesis of RNA. Okay, so here coming to this point, Francis Crick proposed central dogma of molecular biology. Okay, so Francis Crick proposed, so he proposed a central dogma of molecular biology. So this is important. Okay, the Francis Crick who proposed central dogma of molecular biology. So what is the central idea of molecular basis of inheritance is that, okay, from DNA RNAs are going to form, and these RNAs will help in the synthesis of proteins. So the information present on the DNA is transferred to RNA, and from RNA the information is used to synthesize proteins. So this is what actually called the molecular, central dogma of molecular biology. So I will mention this one. So here in detail, so DNA will replicate by itself. So this process is replication, and the DNA will help in the formation of mRNA. This process is called transcription. It's called transcription, and the mRNA will be translated. So the process called translation, and that translation will help in synthesis of protein. Right. So this is the molecular, central dogma of molecular biology. It's called central dogma of molecular biology. Right. So it's important. The DNA replicates by itself, and also DNA will be giving off to mRNA. So here the M stands for messenger. So what is all that I told you already, I'll be discussing in coming sessions. So that's what the DNA depends on RNA for synthesis of proteins, whereas The RNA, you can see that directly according for synthesis of RNA. So these are the differences that exist between, okay, RNA and DNA, and it's the RNA that evolved first, okay, and that is the first genetic material, and the second genetic material is DNA, right? Okay, and DNA evolved from RNA by some, okay, structural and chemical modifications, and it became more stable than RNA molecule. Right, I hope it's clear. With this, I'll be ending today's session. So we'll see in the next session.